Kara Wells, I am so happy to see you again and have the opportunity to talk to you a little bit about the embryonic viability journey that you've been on. Before we get started, if anyone has questions during Kara's presentation, please put them in the chat box. I will try to mute any ambient noise that might try to trickle through here from time to time. Obviously, my name is Andrew Rose. You can see it there underneath my name, and I'm excited again to have Kara here. I followed Kara's journey for it's probably been over 2 years now since I was first put in touch with you after you came through the Grow New York Cornell incubator accelerator, and then onto the Farm Bureau acclaim, and now look at you, you're almost on top of the world at this point in time. And I've asked Cassidy Johnson to join us as well. I think it's important that we have someone that's an actual rancher that oversees these types of animals that can opine from time to time. And I think we just lost Cassidy. So, Kara, at this point, I'm going to turn the microphone over to you and your team. I would love to, first have introductions to your team, but also, what was your journey? How did you get to this point? There's so many different things that a person can do with their lives, and why was it this? And then talk a little about where you are right now. You know, all the things you've gone through to get to where you are, and then what the future might hold for us as well. Thank you.
Absolutely. Thank you so much for having us and the team here today. So my background took some turns to get to where we are today, but I did not grow up necessarily with an agriculture background. I'm from the suburbs of Houston. I'm from an oil and gas family. But as a kid, I randomly became fascinated with horses. I begged my parents to buy me one. They did, and I wanted a career working with them. So I went to college at Texas Tech University for animal science. And that was really when I first got introduced to opportunities in agriculture, how much science really goes into producing all the food, the animals and the plants that we eat. And I kind of on a whim decided I wanted to do undergraduate research. And so I started working in a reproductive physiology lab. And the lab that I was in, the professor actually had dual appointment as the lab director of a human IVF clinic and also at animal science. So I got exposed to both the world of animal and human assisted reproductive techniques and IVF, and I absolutely loved it. We'll get to this in the presentation, but despite all the cool things we can do in advancements, it's still a relatively young science. I believe when things are young science, there's more opportunity to move the needle. I stayed on for a PhD in animal science with a concentration in reproductive physiology. I graduated. I had no idea what I wanted to do. I had the good fortune of sitting next to a woman on an airplane. She's now the chief innovation officer at Tulane University, but she convinced me to start a company and commercialize some research and everything. And so I never saw myself necessarily as being entrepreneurial, but apparently I'm easily persuaded. And so that was back in 2017. And 6 years later, here we are. I'm still an entrepreneur. We pivoted and made a lot of changes since that first company ideation, but I love combining science and actually applying scientific innovation to commercial solutions that can actually help the people making our food. So that's my background.
Not to interrupt you, but you also have patents. You know, I'm a person that I could never fathom in my life ever having a patent, but you have plural patents, don't you?
We have 8 US patents, 7 US patents pending, and 40 international applications. So we've been very aggressive on the patent front. Two of our business partners are actually IP attorneys, which is how we've been able to afford to have so many patents. But, you know, that's something that's definitely been very new to me is sure, you have a good idea, but how can you protect that idea, make sure that you own the IP and not just own it in the US. Our world is so interconnected now. I think the last time we talked was CH4, he was in Australia. And so I really don't think it's smart or practical for a company just to think within the country that they originate from. So we really try to position ourselves for international and global operations. And even if we're not doing those operations ourselves, we can position ourselves for an exit from a large animal health company with those global networks. So yes, patents have been a huge part of our business strategy and something that we definitely invest in to protect ourselves.
And for those of you who don't follow Kara on LinkedIn, she's very proud of these patents. And from time to time, she'll put the technical details up there. And not that I really understand it, but I do support it.
And, you know, I like people to know that we have them, you know. But yes,
It's certainly good for defense, that's for sure.
my thoughts exactly. Yes, you know, I don't want anyone to claim that they didn't know. So I feel like if we air out everything, that if you don't know, then it's ignorance. So that's a personal strategy. I'm actually not sure that my patent attorney agrees with it, but that's my personal strategy.
Indeed. And I noticed that you've got a few friends here on the webinar as well. I mean, you know, I certainly met Russell, but who's your team and what are their roles and responsibilities?
How about I let them introduce themselves? Russell, do you want to go first?
Sure. My name's Russell Killingsworth. I'm a veterinarian from Shamrock, Texas, up in the Panhandle. And I've been doing conventional ET work in my practice for almost 40 years. And we met Kara a couple years ago. We got started on this journey together. And it's been fascinating to be involved in this company. It has been truly amazing.
He's humble. He's really
Yeah, I was going to say that's probably the most humble introduction
kind
ever.
of the brains behind this operation. But Cameron, want to introduce yourself?
You bet. So I'm Cameron Hayden. I'm also with Team EmGenisys. I got hired on last fall to serve in a veterinary liaison role and also serve in an industry relations role. A little bit about my background. I grew up in southwestern Michigan on a show cattle operation. Spent a little bit of time in Kansas for my undergraduate degree, and recently finished up a master's degree in bovine reproduction, and finished that last fall, and I've been with EmGenisys since.
Wow, that's certainly a nice pedigree right there. And Kevin, I know that you weren't expected to be called upon, but who are you and what role do you play with EmGenisys?
So Kara and I met at the Houston National Cattlemen's Convention because Andrew noticed that both of us were there and we should meet. And so Kara and I just kind of bounced ideas off each other, and we come from a seed stock cattle background, Hereford and Charolais, and we utilize embryo transfer extensively because of we focus mainly on our top females to be able to grow that out. And so that's why this technology was of interest. And so just kind of was a fellow entrepreneur in support. And then last fall, talking with Kara and helping with marketing and strategy to get the technology and the process out to the industry as well.
And you're also an entrepreneur, aren't you?
Yes. Yeah. I guess a serial entrepreneur. Dove off into the tech side instead of just the practical entrepreneur with an ag labor app, Ag Butler. That's how Andrew got to meet me at the Ag Innovation Challenge. And then we told Kara that she should apply for the Ag Innovation Challenge, and she was runner up.
She certainly was. And I was telling Russell before she got on, I don't think there's a cardboard check she hasn't held yet in the Ag Innovation sector. If there is, we'll find it and she'll be holding on stage, I'm sure. So Kara, back to you. Sorry, go ahead.
I was just going to say, I think everyone that I work with, they wear a lot of hats. Sometimes they're detectives. They're also awesome at doing market research and figuring out who do we need to talk to, how to solve this. And I appreciate them all for putting up with me. I joke with Cameron that I made him become a camel expert almost overnight to do a project at one point in time, as well as look at other oddball sectors, space ag for one. So everyone's super cool and very innovative minded, and really no one knows any bounds of innovation, which I think is really cool that I get to work with this team.
Well, not only that, it's the idea and the mission of what you're doing. I mean, they're perfectly suited to support you in going along that journey as well. Well, let's talk about embryonic viability. I mean, prior to you coming on the scene, how could you tell if an embryo was viable or not? I mean, I'll let you get into all that part of it.
Sure. Okay, let's see. Can you see a full screen? Okay, great. So I do want to talk about embryonic viability for sure, but I think just to get everyone on the same page here, we'll do a brief overview. And so I'm not just the only one talking. To make this flow a little bit nicer, I'm going to ask for Dr. Killingsworth and Cameron to come in from time to time. But first off, we're going to talk about assisted reproductive techniques today, but specifically the big ones that we mean is embryo transfer or superovulation and in vitro fertilization. These are different processes, but the result and goal is to get embryos outside of the body that we can handle and transfer into surrogates. For superovulation, this is a process where the donor animal, the animal that the embryos will be coming from, she will be set up with different hormones that makes her ovulate more than one egg. If you think of a cow, they're not litter bearers. They only have typically one calf. And so with these hormones, we can make them ovulate more eggs so we can get multiple embryos. She's not capable of carrying themselves, so it is the role of a veterinarian to actually flush those embryos out of her. Once they're collected, they can either be frozen for later use or transferred into recipients or surrogate. IVF is very similar, except for in IVF, we're not collecting embryos from the donor, we're collecting eggs. So these eggs are actually aspirated directly from the ovary, and then they're fertilized in vitro or outside of the body with sperm cells. So we introduce sperm cells to the dish, they're allowed to swim into the eggs and fertilize them. We grow the fertilized eggs, which are now embryos, for about 7 days. And then we get an embryo that's at the same stage here that can also be frozen or transferred into recipients, as we call them, or surrogates. And so the ability to handle and have embryos outside of the body really creates a ton of potential for how we can breed and manage different animals. The most common reason that we'd want to go through any type of IVF or embryo transfer is to perpetuate the genetics of the superior animals. For many decades now, it's very easy to perpetuate genetics of a male embryo because they're not actually carrying those offspring and gestating them. We can breed our males to several females each season, and his genetics can be perpetuated. But females also can have great genetics. In nature, they're bound to only having one offspring per year, but with embryo transfer and IVF, we can put their embryos into surrogates and we can perpetuate multiple offspring from these superior females each year. We can also have the opportunity to freeze or cryopreserve these embryos. The ability to freeze them means that these embryos can be maintained viable for theoretically an infinite amount of time. This allows us to store these genetics for later use. If we're thinking about a endangered species project here, we can freeze the embryo of animals that are alive on this planet today and store their genetics so we can supply genetic diversity for the future. We also can freeze them so they can be shipped around the world. This is a big issue right now with all the food security concerns, especially in more third world countries. If US dairy cows produce more milk than the average animal, we can ship our genetics over to these countries and help them have more efficient, high producing animals so they can feed more people per a single animal.
Hey, Kara, quick question for you. What are some of the hot genetics right now that people are seeking out? I heard about the slick cows. Is that entering into this at all? Or are there other things too that we should keep an eye out on?
Sure. So the SLIC gene is definitely important because a lot of these regions that are really suffering food security crises, like Africa, East Asia, they're very, very hot. And so a lot of these high producing European type breeds that are more cold natured, if you will, sure, they would be a great source of meat or milk for these countries, but not if the animals just have heat exhaustion and heat stress and can't thrive there. So abilities like the SLIC gene to make these animals less hairy keeps them cooler and they do well. Really the goal is, I've seen more demand on the dairy side than on the beef side, but they're both in demand. Areas where they don't eat beef, like India, because of religious purposes, they're not so much interested in beef, but they're definitely interested in dairy. We've worked with a group that created the first Western dairy in India, and we're working with other partners as well to see how can we make these more Western genetics thrive in these regions. And years ago, the thought was, sure, let's just send them live animals. Well, there's a lot of logistical concerns with shipping large animals from the fact that sometimes they're just not that cooperative. And then also quarantine restrictions. So there is regulation around shipping embryos, but embryos are about the size of a grain of sand. They're a lot easier to ship, and there's definitely less regulation than shipping live animals. Different assisted reproductive techniques have actually allowed us to sex sort and control the sex ratio. So if you're dairy, you want females to make milk, we can turn the odds in their favor where they can have over 90% of calves born be female without the culling of any males. Some groups, they might want steers. So we can have different technologies that really allow producers to choose the sex that they want born. So it's more economically and advantageous for their systems. I'm sure you've heard about gene editing. The SLIC gene is part of this. Since CRISPR technologies really advanced, there's a lot of different companies trying to look at gene editing, whether it's to edit out diseases, make animals more productive, or just have traits that make these more desirable. We're familiar with some gene editing groups that are also using gene editing to control sex ratio. So a lot of these categories that I mentioned really do intermingle, but without the ability to get embryos, gene editing wouldn't really be feasible. This also goes into cloning and stem cell research. Cloning is definitely more reserved for like the elite genetics end at this point, but it can also play a role in species conservation as well as stem cell research. So there's definitely advancements being on this. And I put posthumous reproduction on here because now an animal no longer has to be alive to be able to reproduce. If people intervene soon enough, we can still collect gametes, egg and sperm from these dead animals, or even somatic cells like cartilage, ear punches that can be used to clone these animals. I gave this presentation to a wildlife group recently, and this is definitely of interest to those groups because cows are pretty easy to catch. We can herd them into a chute and do whatever we want to them. Not the same thing can be said for wild deer and exotic species. If there's a animal who dies, we can still save their genetics, and it might be easier to harvest the material once they're dead than alive. So a whole world of opportunity is really available here when we can do techniques like IVF and embryo transfer to handle and manipulate embryos outside the body. And I think what I'm telling you sounds very Brave New World-ish, but it's actually been around for a long time. I'm not going to go into the total history, but artificial insemination has been around since at least 1322, likely sooner. Arabian chiefs would steal semen from their rivals and use it to inseminate their own horses. The first embryo transfer was done in 1891, and rabbits, but it wasn't done in ruminant species like sheep and goat until 1934 and 1951 in cattle. The first time an egg was fertilized in vitro was in 1933, even though it didn't result in a live birth until 1959. Frozen embryos have been around since the first calf named Frosty was born in 1973. And I put Louise Brown on here because this was a major global disturbance. This is the first human IVF baby born in the UK in 1978. And after her birth, Congresses around the world immediately went into session to see what we were going to do about this crazy phenomenon. I think last year there was over 2 million babies in the US born from IVF. So things have definitely changed. Sex semen has been around since 1989, and it was brought to cattle in 1993. And the first clone was made in 1996, which is Dolly, named after Dolly Parton, because she was cloned from a mammary cell. So there's a little humor in that. And I also, since I'm an entrepreneur, I wanted to put some of the corporate groups in this timeline as well, because these groups are really responsible for bringing these technologies to the masses. Some of these companies marketing semen have been here since the 60s. ABS was created in 1941, and it combined with in vitro Brazil in 2002. Transova, who is definitely a leader in cattle IVF as well as other species, has been around since 1980. ST Genetics was created in 2004 to bring sex semen to market, and they still have been very prominent in their patents, doing that process today. And then in recent years, more groups, I definitely did not include all of them, but Urus emerged in 2018, Viability emerged in 2015, and I put Colossal up here because since 2021, they've raised $225 million to de-extinct the mammoth, dodo bird, and Siberian tiger. So things are happening.
Wow. I mean, going back to species preservation, it sounds like that could be an angle you're exploring as well.
I mean, I think that we could definitely add value. I don't think anybody, even the folks with millions of dollars at Colossal, know how to take care of a mammoth embryo. So, with our technologies to help give more objective data, I think we can definitely maybe not solve their problems, but give them clues to better solve their problems faster. But, if you look at this timeline, the last advancement here was 1996. And what's been going on since the new millennium is more of a commercialization, bringing these services to market. But not a lot of innovation has really transpired in this industry since, my career started and, since Dolly was born. And when we look at pregnancy outcomes, we kind of see the same thing. So this is not my data, and I don't want to spend too much time on this, but some of these graphs show in vivo and some of these graphs show in vitro fertilization. But if you look at this, almost all the numbers are below 50%. This has been since year 2000, and we haven't really seen any improvements in getting more animals pregnant as a result of these procedures. Russell and I had some fun one day, and we went back and looked at his year-to-date outcomes since he's been practicing in the 80s, and he's been around 55% successful in conventional ET for the past 40 years. So there hasn't been a innovation, technology, or advancement that increased the spike of pregnancy outcomes. And this is really bottlenecking our ability as an industry to scale these services into the commercial sector and really reap the benefits that the perpetuation of these genetics can cause. So here's my whole platform is you might have the most expensive, valuable animal. It might produce more milk. It might always grade prime. It might, be amazing, but it doesn't really matter how genetically amazing that embryo is if it's not competent, viable, and capable of making a pregnancy. if it can't result in live birth, then we shouldn't be using resources on it because it's valueless. And so last year, we know that there were over 5 million cattle embryos made in the United States. About 2 million of those were transferred. But like I said, we've been stuck at a 50% or less pregnancy outcome since the 1980s. In my market research, we've identified $120 million opportunity just to evaluate embryo viability in North America alone. And this is an opportunity that's growing at a rate of 17% year over year. And the 17% comes from the volume of embryos that's made year over year. As more companies like Transova, Bovatech, Vitelli, Simplot, Hoofstock Genetics start, actively marketing the sale of these technologies, they're making more embryos. And so that's more embryos that need to be evaluated. And then globally, this is a $20 billion market, and that does include the use of this for clinical human IVF. And this is all great. We can make a lot of money. As scientists, we can definitely solve this problem. But something that I've definitely run into is my experience as a scientist, as an entrepreneur. And if we had more time, we could talk about some of my failures as an entrepreneur and lessons learned. But it doesn't really matter how accurate and scientifically credible a technology is if it's not practical and doesn't fit into the workflow of the rural veterinarian and livestock producer. So when we go to make new technologies and innovations, at the forefront of our mind is, are they practical? Are they non-invasive? Are they objective versus subjective? And can they be scalable to be used on just a handful of embryos and also at large volume laboratories? And I don't think there's anybody else more qualified to talk about the needs of the practical rural veterinarian than Dr. Russell Killingsworth himself. So Russell, I'll control the slides if you want to let me know when to proceed.
Okay, thank you. Thank you all for having us today. Once I'm able to collect the embryos from the donor, I have them in a petri dish under the microscope. I get them gathered up in a group, and then the real test comes in. Can we pick out the viable embryos to transfer into the recipients? And to date, we base this simply on how they look. Do they have, are they at the right stage of development relative to the time of ovulation, the number of cells, are the cells uniform, color, homogeneous in their appearance? But all of this is a subjective evaluation. It simply depends on my experience, my equipment, and there's nothing objective about it, nothing to measure other than, does the embryo look like the pictures say it should look like? And the problem that this fails to identify, what research has shown, is that up to 20% of these embryos are non-viable and have no chance of establishing a pregnancy, even if they appear normal. And when we think that this 20% that cannot establish a pregnancy, that results in almost $2 billion in cost to the American cattle producers each year. Now, okay, when we look at an embryo within just the first 7 days of development since fertilization, we know that embryo has changed from a single cell fertilized zygote into a cluster of 150 to 300 cells blastocyst. Those cells have differentiated into the outer trophectoderm that will become part of the placenta, the inner cell mass, which will develop into the fetus. But we see that these cells are growing, dividing, changing, rearranging. The embryo is a dynamic process. Things are going on all the time there. And so that's what our technology is trying to look at, and to identify these changes that are taking place. Okay. Especially in the human IVF processes, time-lapse imaging has become a big deal there, where they take these embryos are in culture in the incubator, and they record an image every few hours and record the changes. And through time, they have been able to identify some particular events that have to take place at this time of development in order for this embryo to continue to develop. These incubators with a microscope in the incubator so that you don't have to handle the dishes, these are extremely expensive systems, not really practical for livestock application. Also, these only work for the in vitro embryos that you're going to watch develop for 7 days. In my practice with conventional embryo transfer, I only get one look at that embryo at day 7 when I get that embryo from the cow. I've got to make my decision right then on, is this a good embryo or not? Can I transfer it? This journey started when Dr. Wells saw a TED Talk video on motion magnification. A group at MIT, engineers at MIT, had developed a software that was able to take a video, and then using algorithms, the computer was able to pick up on minute pixel differences frame to frame. And then when they magnified these differences, it made it become visible to the naked eye. The top images there are just still shots taken from the original video. Once they process this video through their software and pick up the differences in pixels, you get the still shots in the second row, where you can actually see color changes in the skin with each heartbeat as oxygenated blood is flowing through the tiny capillaries in the skin. So again, we can't see this with our naked eye, but the computer can see these differences. So Dr. Wells called me and said, "Do embryos move?" I said, "I don't know. Let's see." So I had a camera. We took a video. We bought the software. And then when we processed this video through the software, we came up with this, and things did seem to change and shift and move and activity within the cells. So this was exciting. And initially we thought movement has to be good. If it's moving, that's got to be good, but we didn't know how much was enough or is something too much. So again, this process needs to fit into our everyday workflow. So as I collected embryos from donors in my practice, I would record the videos of those embryos, then we would transfer or freeze those embryos for later transfer. And then I would send those videos to Dr. Wells. Dr. Wells began to actually do measurements on the videos, where she'd measure the X and Y diameter of the outer zona pellucida of the cell mass itself, then take measurements from the cell mass to the zona, all points around the clock face. Then we'd move the video 5 seconds. She'd take the measurements, repeat them all over again. So we had spreadsheets full of numbers, but we didn't know what it meant. And then when we started getting pregnancy data back on those embryos that were transferred, and we put that pregnancy data against the movement of those embryos, we get this nice bell-shaped curve where we see that the embryos with the more moderate movement have your highest conception rate. Those outside one standard deviation. Those that are too active or those that are not active enough, you have lower conception rate. And so this was really exciting, and this is what got us started thinking that we may have something here. There's some other work that's been done by Dr. Lisi and some of his staff, where they were actually measuring metabolic activity of these embryos based on pyruvate consumption. And they developed what they call a Goldilocks range. Embryos that had metabolism that were too high seemed to burn out and did not establish a pregnancy. Embryos with metabolism too low could not support life and did not continue to develop. But it was those in the middle, as far as metabolic activity go, that got the most pregnancy. And that fit very well with the data that we had seen. And so again, this was confirmation. Are we looking at actual metabolism in these cells, this activity that we're noting? But to be practical, there's no way to take the time to measure and do all that Dr. Wells had done looking at each video. This needs to be able to fit into my workflow today. Of course, we can't do anything that will be damaging to the embryos. And so we started looking at recording these. This is just a simple setup with my smartphone on the microscope, showing how we record a video of these embryos. And then we've developed software where we can upload that video, and then it is analyzed. We went to Texas A&M, to the biomechanical engineers, because we knew that there had to be a way to automate this system, to do it in real time if we were going to make this work. Now, on the left is an actual video that is playing. The middle video, you can see those changes. That's the same video as the embryos on the left. They're using a process called image subtraction. The computer looks frame to frame. Anything that is different in one frame than the previous frame shows up as a white dot. So those little white spots that you see moving and changing in there, that is change within that embryo from frame to frame. And then applying one other filter there on the right with those colors, we can actually get a direction of the motion based on which color. And also the intensity of the color talks about the magnitude of the motion. So a lot of information there in that embryo that is changing, going on in this living mass of cells. Okay, Karen. Next, we needed to make sure that what we were seeing was in the embryo and not external noise due to fans, motor, frequency of UV lights. And so they would take a box of the action and there in the embryo, then they compared that movement with four corners outside of the embryo to compare that movement. And then the graph there on the bottom, the top squiggly blue line represents mean motion of the embryo itself, the action region. And then the motion of those four corners outside of the embryo are represented by that bottom line. That's all four colors right there. And you see that the activity, there's no change outside the embryo. The change is all occurring within the embryo proper. So again, more validation that what we're seeing is due to changes within the embryo itself. Okay. We have looked at this. Again, we started looking at this with my conventional ET work. We've also looked at some IVF embryos. We went to the Reproduction Service Center there at A&M. They did a big in vitro study where we they produced embryos, cultured them. We recorded videos, ran the videos through our process, and made our predictions. And we used that hatching of those embryos as the end point. And so that's some of the early work that we had done there with A&M. And in this study from A&M, the embryos that hatched and were viable, we see that the range of motion is much lower there on the left compared to the non-viable. And also when you look at the mean, this is statistically significant, the difference in the amount of activity between those that hatched and those that did not hatch. And so this has led us to what we think we can do here. Here on the next slide, that we do have a system that's non-invasive. We're able to do this in real time as we record a video, upload it to the computer system, and it can run the algorithm and print out a report showing us a viability score for those embryos. And if we can eliminate those embryos that have no chance, we're not going to waste time. We're not going to waste recipients on those, and we can actually increase pregnancy rates of those animals by putting good embryos in all of the animals. And so that's where we're trying to go from a practical side.
Dr. Killingsworth and Kara as well. Keith just messaged me. He's got a question he would like to ask before he has to break away from this. So Keith, I'm going to turn the microphone over to you at this point.
Awesome.
Well, thank you. Everybody can hear me? I
Yes.
appreciate you all letting me join you today. It looks like I'm probably the only non-scientist here, or non-vet. I haven't met each of you, but just so you kind of know where I'm coming from, I am responsible for the fresh meat operation at a grocery store chain that has about 1,400 stores. So we buy a lot of meat and we sell a lot of meat, and then we answer a lot of questions from regular consumers because of that. So really the only frame of reference I have, I can't debate you on the science, but I think where our worlds intersect is potentially that I have to be a science communicator and you all have to be scientists. So I jotted a couple of things down because first, you were talking earlier, Kara, about promoting your patents. Great, right? Then I think a couple of words caught my attention, that posthumous reproduction, and I could kind of see that getting out, or the everyday consumer hearing that phrase and other phrases like it, or potentially an NGO of some sort that's kind of antithetical to the meat world, using that to kind of foment some fear and something. So I don't know, from the business side of what you do, what are your plans or your intentions to kind of manage all of that, so when this does get out, folks like me can communicate it the best way possible when we get the inevitable emails that are we using CRISPR, or are we using gene edited animals? And to kind of put it in perspective for you, over the last month or so, it's tapered off, but we were up to maybe 30 or 40 emails a day from customers about mRNA vaccines and wanting us to label our meat. We're not using that. So it consumes a lot of time, and you want to be kind and caring to folks to communicate it. So I don't know what your thoughts there on, as this becomes more commercially viable or becomes more well known to the public, from a business perspective, Kara, how do you plan on managing all of that, or how does it get managed?
Sure, those are definitely great questions. My cousin actually texted me yesterday asking and being concerned about mRNA vaccines and livestock. So I completely see where you're coming from. I think something that my team does exceptionally well, as we don't operate in a vacuum. I know Cameron, before he came onto EmGenisys, he really calls himself an advocate, where he's trying to advocate for ag outside of the ag community and make consumers feel more comfortable with the foods that they come in their body. When I talk about posthumous reproduction, I really am not thinking or promoting those abilities for the food animal standpoint. That's more like if a hunter shoots a prize deer. For example, one of our clients owns the world record kudu. It's the biggest one ever recorded. He's 12 years old, and they're concerned that his life might not be much longer. They would like to breed him after this, so they're taking efforts to see how can they preserve his cells. Another big important one that has been in the news for posthumous reproduction is the northern white rhino. And I think this is a really good public feel-good story. Right now, there are only 2 northern white rhinos alive on this planet. They're both females and they're both sisters. A few years ago, Sudan, the last male rhino, he died, and they froze almost everything that they could have off of that male rhino. There's been millions of dollars that's gone into this project, namely at the San Diego Zoo, where they're trying to do these stem cell projects to repopulate the northern white rhino using southern whites as surrogates. So the ability to do posthumous reproduction on that can keep these animals on our planet longer. And so I think from a public relations standpoint, consumers resonate with a story like species conservation, such as with the northern white rhino, more so than they would want their food coming from dead animals. And because you and I both know economics matter, I don't personally believe that there's a huge economic advantage for doing posthumous reproduction on beef and dairy cattle. So I don't really even see that as an economic driver or a market in that in the first place. 2, what you're saying about public concern, GMOs, that's absolutely a concern. It's a concern that we treat with a lot of respect. I hope you watch the rest of this presentation when you get the chance on YouTube, but you'll see that all of our analysis is just on video data of embryos. So we're not actually doing any manipulations to these embryos that aren't already being done. Our company only processes video data. We don't actually process the actual embryos themselves. But I think from a public relations standpoint, when we can say there's this dairy cow in New York who doesn't need to eat very much, but she produces a lot of milk. This is a great environmental story because she's less drain on the environment and she's able to produce more gallons of milk. Or the steer that produces a lot of lean muscle mass from little grain. If we can breed these animals more that just naturally grow fast and don't need to consume a lot of feed, that means that we're reducing the carbon footprint and the environmental footprint by producing food animals, and our ability to better perpetuate those genetics helps make all the animals that we breed more efficient so we can really feed more people from fewer individual animals. I do have kind of a nice take-home slide with these impacts on it that I like to show the public sector, and I think Russell, Cameron, Kevin, and Andrew does a great job at being an advocate as well, of just trying to show that there's people that care a lot about animal health and welfare, and also a lot about science, trying to feed the people in this world. I think that we all are very committed to that. And so, we're trying to create safe ways for human health and for animal health that still gets the job done of feeding this world, but it does it in a safe and sustainable manner. So, I'm definitely kind of a purist. I don't like my food too manipulated either. So I get where these public groups are coming from. But I also think that helping educate them and showing how much care, precision, and science is going into their food should help create some confidence in these food systems, and hopefully makes people feel good about the products that they're buying from American cattle and dairy producers.
Well, thank you very much.
Absolutely. Thank you. Do you have any additional questions?
No, I'm good. I'm gonna, I think you answered most of them. Most of them were just about the science communication part of it. And I think that that's just a critical component of making sure that these technologies are not rejected by the general public.
Absolutely. And then when we bring it to something that people are very familiar with, human IVF, Luckily, almost everybody, if not themselves, well, not luckily, but from an education information standpoint, most people are very familiar with the IVF process because someone close to them has gone through that. They know what their embryos went through. They know that they have normal, healthy children because of these processes, but they also hate the fact that their doctors might have put dead embryos in them and charged them $60,000 to do so. So they understand that technologies to help select healthy embryos can improve pregnancy outcomes and, more importantly, live birth outcomes of these processes. And so whenever we are conveying scientific information, I do use human IVF examples a lot because people are just more familiar with them.
Thank you. Thanks for letting me join you all today. I appreciate this time and getting to learn a little bit.
Great, thank you. Well, Cameron, do you want to start over from the beginning of your presentation?
Yeah, we'll start from here. And so, as we continue through, Dr. Wells, Kara shared quite a bit about the advancements in assisted reproductive techniques, and Russell shared how, from a company standpoint, our initial product is based on evaluating embryo viability. And throughout this process, that we have really realized that there are tons of variables present within embryology as a whole. For instance, different species have different embryo production rates or speeds, and those embryos behave differently. And even within breeds or within species, different breeds, whether that's beef versus dairy cattle, their embryos will behave differently. We know that there are environmental impacts of various temperatures and different locations that cause development in embryos to change. We also know that nutrition, stimulation protocols, and animal age on the donor side all have impact on embryo production. A big difference, a variable, I guess, within the industry is production type. We mentioned conventional embryos versus IVF or in vitro produced embryos, and those embryos have different developmental rates and developmental speeds, by nature of that being in vivo versus in vitro. Also, some hardware differences in cameras and microscope equipment will cause variation in how those embryos appear to be graded. And then finally, technician experience really has an impact on how I may view and grade embryos versus Russell, for instance, who has much more experience. And so when we take all these variables into consideration, it definitely makes it a challenge to evaluate embryos, under the microscope with our eyes, but also it provides a challenge for machine learning and technology to really take some of those variables out of this process. And so here, as we continue to the next couple of slides, it's some preliminary data that we have captured within our company. And as Kara mentioned, all of this research and data is presented on just 30-second videos of embryos under a microscope, so they haven't been manipulated or handled, and it's a very non-invasive process. And so as we continue, I think our first study here would be done on conventional embryo production in the Texas Panhandle. And this is all based on the collection season of the embryos from the donor. And so here we have in blue would be donors that were flushed, and those embryos were conventionally collected in the summer months. So those donors could be at the potential to have heat stress, versus in the orange, we have donors that were collected in the winter months like December, January, and February, where we would assume based on temperature and climate that those donors would not be subjected to heat stress. And so on this graph, we can see on the y-axis, we have the standard deviation over the mean of pixel change, and that goes back to the process that Russell shared about the pixel change and differences frame by frame of those videos, specifically of each embryo. And in this particular study, we can see that those embryos that were collected from potentially heat stressed donors there in the blue had a greater mean change or morphokinetic activity than those that were collected from non-heat stress donors. And so this is a testament to our technology being able to identify some differences in embryo production that ultimately lead us to believe that those embryos produced from those cows in the summer months would have heat stress. And as we know from an environmental standpoint and production systems throughout the world, if those donors are heat stressed and they're producing embryos that are heat stressed, they're likely to have a lower pregnancy when transferred. And so by having some preliminary data on this, it proves that we might have the potential to select heat stress embryos and eliminate those from transfer as well. In our next study, this would be a sample size of 95 embryos collected from Holstein donors, also conventionally flushed. And so to get orchestrated with this graph, we have also the mean pixel change in the y-axis, and then temperature in Fahrenheit on the x-axis. And so way off to the right, we would know that our cow at the time of flush, her internal temperature is about 101 degrees Fahrenheit. And so as we collect those embryos from the cow conventionally, then they're placed into a media dish or into a holding dish with media. And so when those embryos were placed into that dish and videoed, they were also taken a temperature of the media. And so as we can see here, that as we move further away from the cow's natural body temperature towards 70 degrees, those embryos tend to move more and have a higher morphokinetic activity, which we would see as a subclinical stress detection. And if we follow that trend line, we can see that on both ends, so near about 75 degrees and then up near 90 degrees, those are highly correlated. But within the middle there, between 80 and 85 degrees or so, those embryos have lots more variability. And so we did follow those to pregnancy data on this slide here, and we can see that it was interesting within the variable section about 80 to 85 degrees, those embryos that maintained a more quiescent state or had less change achieved more pregnancies than the embryos that had more change within that intermediate section. And so as a whole, this is just interesting that we can leverage machine learning and technology to identify some of those embryos that have stresses subclinically, and then also potentially be able to identify those embryos that, although there's fluctuation in temperature, might be able to achieve a pregnancy when transferred into a surrogate.
Cameron, quick question for you. Would you know that the eggs were harvested from a heat-stressed animal, or is this the first indication that you would have looking through these charts?
Yeah, so in that initial study, we did not track each cow individually. It was a retrospective study based on collection temperature and climate at the time of collection for the donor cow. So not necessarily, we did not check each cow, but based on the climate temperatures in the Texas Panhandle, we would have reason to assume that some of those cows would at least have been heat stressed during the collection period. But there's plenty of research out there that they have taken, samples and biopsies of embryos from heat stress cows, in addition to looking at just the conventional flush records between cows that were subjected to heat stress, and any of those that were heat stress had lower embryo production, and those embryos would, have lower pregnancy rates when transferred.
I read an article, I can't recall if it was Ford's Dairymen or Progressive Dairy, and they did a study on heat stressed dairy cows. And not only does that trauma occur in the animal, it's passed on to the offspring, and those offspring don't do as well as a non-heat stressed animal as well. So this is a critical point.
Yes, for sure. I think I probably read a similar article there on the beef side. There's evidence that calves born from heat stress donors will not achieve, an optimum mature weight or won't grow as quickly as their counterparts that were, born non-heat from a non-heat stress donor. And then also, yes, on the dairy side, from a total milk production standpoint, earlier in their life, that those come from a heat stress background would have a lower performance. And so, yeah, we really think that if we can continue with the data set to identify those embryos that are heat stressed, and of those heat stress embryos, specifically ones that would not achieve a pregnancy, that would save, a lot of money down the road for producers from a practical standpoint.
And I just want to add to Cameron's point, from a morphological standpoint, all the embryos in these studies look perfect. The human eye cannot identify embryos that have suffered from heat stress. So to my knowledge, this is the first non-invasive way to detect embryos that have had stress and be able to detect it subclinically before outward signs of stress and deterioration really impact that embryo's development. So we're excited about this data. It's preliminary, but we're very optimistic about it.
Yeah, great points from Dr. Wells and Andrew there. And as we move into our next study, we did a morphokinetic activity analysis on embryos that were cryopreserved and then thawed. They were thawed and allowed to develop there for an hour to reabsorb. And then we took a video an hour after thawing and 24 hours later after put into incubation. And so we can see here, just from a small sample size of 32 embryos, that the embryos that developed from the initial thaw through the 24 hours later in the incubator, that we can see with our technology had greater development activity as referenced in our blue plot versus those embryos that stalled or did not appear to have any visual growth or change after 24 hours of post-thawing, even in an incubator, were slower in development and had slower activity. And so this is also an indicator that hopefully down the road with some increased data and refinement in our machine learning models, that we can identify those embryos that would have a greater potential to go through freezing and thawing without having quite as much cryo damage or damage from that freezing process. But this is, like I said, just good, interesting data from the start to realize that our technology is sensitive enough to pick up differences in development post-thaw from those embryos that were cryopreserved. In our next study, we have some preliminary data on sex selection. And so it's, I guess, foremost important to note that here on the left, that is a direct image of the video quality that was subjected to this data analysis. And so we can see that's really what we would deem currently as, poor resolution. It's pretty pixelated and dark, kind of hard to see. And so while in this process, we had 61% accuracy using a K nearest neighbor predictive algorithm, which uses machine learning models to take an embryo selection and use a number of neighbors to make predictions on a certain parameter. And so this parameter would be sex selection. And if we look at the chart on the right, we can see our blue trend line was the highest at about 61% accurate for predicting embryo sex when we used 3 neighboring embryos to make that prediction. But like I said, this is very preliminary. We did have about 102 embryos in this sample size, but with the poor resolution in these videos, it makes us kind of excited as a company that down the road, with the data that we are capturing internally and that our customers are also sending us now are much higher quality videos taken right, from their iPhone or cell phone, that we should see a much greater accuracy in sex selection and prediction, non-invasively, specifically from embryo analysis videos. Our next,
Cameron, a quick question about that. when you're looking at batches of embryos to determine the viability, is there a certain number that you examine at once? Could it be 1,000, or is it, you know, I've seen several slides with 5 embryos. This one's got a few more in there. What does that optimal number look like? Or is there one? I don't know.
In our current, released bovine models, we recommend, up to 7 or 8 embryos per video analysis. And that's because based on, the screen size of your iPhone and being able to have those embryos in the screen with optimum, zoom and without those embryos touching or overlapping, that we can fit about 8 in that screen. But we have done, like I said, that's most specifically in the bovine model, but we've also done some data exploration and some preliminary work for other customers that have sent, up to, probably 50 or 60 embryos per video, but it makes it a little bit different depending on what the end result of the analysis is. And so on the bovine model, when we identify, if we have 8 in a screen, we're maintaining individual embryo identity because each one of those embryos will be loaded into a straw to be transferred in or frozen, whereas in some of those other research models, we're doing a little bit more of quality control work. And so the individual embryo identity isn't as necessary. And so you can fit more on screen just from, like I said, a lab quality control purposes. So yeah, we recommend though for customers that are using this for an embryo viability model to have up to 8 embryos per video. So here, I think our next data set, preliminary study we have on cloned embryo activity. And so we think this is actually really exciting. Probably some of the first data out there from the, a short video analysis of embryo activity throughout the entire clone process. And so if we think of this, the biological processes here in stage 1 in the dark blue on the very left, we have just a video of oocytes that are analyzed for morphological or pixel changes. And these embryos show, or these oocytes, show greater variability. And that would really be kind of exciting for us because it's depicting these embryos coming from different oocyte donors that are pooled together. And we know from a biological standpoint that different donors, different oocyte donors will provide different quality oocytes. And then also, depending on when these oocytes were collected or from what follicle size, that some of these oocytes would be going through atresia and no longer viable versus some would be at optimum viability. And so our technology was able to depict that within the stage 1 we have on the left for oocyte quality. And then for stage 2, in the process of somatic cell cloning, we have these oocytes are enucleated, or the removal of most typically the polar body and the metaphase plate. So right now it would be kind of what we would deem as kind of like an empty oocyte. There's nothing there to really trigger it to continue to grow or develop. And so that's why we see a slight decrease in activity levels as it relates to the initial oocytes. For stage 3 here in the middle on the gray box, we have injected oocytes. And so once we, in stage 2, removed the polar body and metaphase plate, now in this specific data set, a new diploid donor cell was inserted into that oocyte. So now we have that in there, but it has not been activated yet. And so from a biological standpoint, it has the parts to continue to develop to an embryo, but we have not told it to continue to develop yet. And so that's why we see in this chart also a little bit lower activity. And then finally, when we go to stage 4, which is post-fusion, so between the gray chart and the yellow chart here, we have an electric process or chemical process that shocks these oocytes and diploids to fuse. And so in stage 4 now, we have these couplets that are fused together. And so we have told them to start and continue to develop. And so that's why we see a jump here. But it's also important to note that there's lots of variability within stage 4 here post-fusion. And that's, indicative of, in the process, some of those will fuse and go on to develop, but some of them will not fuse and will go through atresia. And so we can already see lots of variability within this chart specifically. And then here, our lighter blue is stage 5, which is pre-ship. And so this is where they're developing into some of those are developing into embryos that will go on to be transferred into surrogates. And then once again, some of those that did not fuse would be going through atresia as well. And then finally, we can see that there is a little bit more activity here in what we would deem as, stage 6, in this case, an oil test, but that's just a continuation of the development from, stage 4, stage 5, all the way up to stage 6. And so this is really exciting data for us that our technology can detect these biological differences and reason through this all the way from the oocytes through the blastocyst development in a cloned embryo process.
Cameron, quick question. I have no idea what an oil test is. What is an oil test?
So it's basically the oil is used, within the IVF system to provide kind of a protective barrier between the developing embryos and the environment as a whole. I'm not super familiar with the entire process. I'll let Carol or Russell share a little bit more if they'd like.
So I'm actually kind of in the same boat as you, Andrew, as I didn't get a lot of background knowledge, but just as someone who has worked with a lot of IVF embryos together, I think this was an internal quality control test that this group was doing. So at stage 5, like Cameron said, these embryos are packaged up and they're ready to be shipped off and be transferred into that surrogate animal. Stage 6 here, they actually grew the embryos past the shipment stage because I think they wanted to test which embryos were growing and which were not. So I would screen share, but I don't want to mess up my slides and everything by pulling up the wrong thing right now. But if you looked at this video, I believe that it was a mix of embryos that stalled in development. That was very obvious because they were small and degraded. And then there's some embryos that made some very nice blastocysts, which is what we want to see. In this particular study, 37% of those made blastocysts. So I think this client was able to say like, hey, yeah, we only had a 37% development rate. Why wasn't it higher? So this oil test that they did, I really think that this was one of their quality control tests internally to see how are their embryos developing, because once they're transferred, we can't look at them, right? So sometimes it's nice to keep some of them outside of the animal and look at them just to make sure that they're developing. And what we're working on today is to separate those embryos that made blastocysts in this test, which I assume would be the faster ones because they have about 150 cells that are active, compared to the degenerates, which I would assume would be the slower ones. So I think we have more opportunity to further tease out information in this. And this is brand new data that we got in this week. So this is the first time it's ever been shared.
Oh yeah, I see the unpublished now at the bottom.
Well, good. This would lead to our final kind of comparison of data here would be a comparison of embryo activity from 3 separate practitioners or practitioner groups. And we can see here for group A has a very large variability within the change and pixel change that we're seeing, morphokinetic activity that we're seeing from their embryos. And when we compare that to group B, which is very tightly controlled, there's not much variability, not much difference in the morphokinetic changes. And then we have group C, C-22 would be data from a practitioner customer during the year 2022. And a follow-up, group C23, would be the data that we have currently from this practitioner throughout 2023, throughout this year. And so we can see in this chart alone how much variation and difference there is between practitioner groups, but also that for most practitioners that utilize the technology year after year, there is some holdover and some consistency in their results from, in this case, group C 22 through 23. And ultimately that, if we go to our next slide, Kara, ultimately we have all these variables that I addressed at the very beginning, and I shared, a lot of the studies that we have right now from a preliminary standpoint that our technology is able to detect. And, so what does that mean at the end of the day? And that means that we have really realized that with all these variables present and different goals for different customers, that customization of machine learning models and customization of our technology to specific customers is how we can increase and have the greatest accuracy for each customer. And so what that looks like is a process where we have data sent to us, from specific labs, partners, practitioners that can be trained specifically to their outcomes. And so in some cases that looks like quality control data. In some cases that looks like pregnancy, optimum embryo viability data. And in some cases that could also look like, embryo sex data. And so once we have that data collected, we test models on our end and refine these machine learning models internally, so that when we have a model release for a customer, that it will, have the optimum accuracy that's available for them for whatever process that they would like to perform. And with that, I think, Kara, I will turn it back over to you for a continuation of the presentation.
Are we good on time, Andrew? Do we have 10 more minutes? Okay, cool. So Cameron and Russell both did an awesome job at sharing a machine learning method that allows us to know what we're looking at. But because we're not satisfied with just one good test, I'm going to tell you about a second test that we apply to our data, which is more of a deep neural network, convolutional neural network method of machine learning. So I think what Cameron and Russell described really does a great job how we can look at an embryo's activity based off video data. But there's actually more data, as you can imagine, in these videos that we can also extract. So machine learning, artificial intelligence, computer vision, these are huge buzzwords right now. Tremendous advancements have been made in the past several years, allowing a group of animal scientists like us to actually utilize these very advanced concepts to applications of our choosing. But just in the most simplistic terms is, what is machine learning? So I think of it like this: computers can learn anything, but like a child, we have to teach them before we can expect them to learn. So in this example, I'm telling the computer, this is a circle, this is a square, this is a circle, this is a square. This is labeled data. It's the shape with the name of the shape associated it. The next time that the computer sees this, it says, oh, I was taught this is a square, and it can successfully predict that. Oh, I was taught this is a circle. But then I threw in this little square with rounded corners, and it's never been trained on something like that, and it gets confused. This is why we need lots of data so we can show the computer every type of embryo under the sun. So when it sees something like that, it can accurately make a prediction and be robust enough to learn to these different things. So how did we do this? And how do we take this beyond circles and squares? So if we think about a video, it's really nothing more than a bunch of pictures, still photos stacked on top of each other. We call these frames. So the average iPhone settings, you can change them, but they're typically set to record videos at 30 frames per second. So this would mean that a 30-second video at speeds of 30 frames per second would have 900 total frames. This gives us 900 pictures per test to train and test our models. So at first, we couldn't train our models until we had pregnancy outcomes. So we took all the embryos that we had, we transferred them into recipients, and then we used their actual pregnancy data to train their models. So if we knew that this embryo went to recipient A and it made a pregnancy, we could train the model with that data. And using pregnancy outcomes really voids us of human bias, because we're not making a human say, well, yes, this one looks good. No, we're actually training it on pregnancy outcomes rather than a human's assessment of that embryo's quality. So this method took over a year of really just training models. So we had basically just dumped them into the computer with the labels. And then there's a lot of different models that we can use, and different models being released every day. But essentially what all these different models do is they look at millions of parameters per frame, and they create these very, very large, complicated algorithms with many, many, many variables in them. And that can be a predictive algorithm. But really what they're doing is they're learning to recognize patterns associated with embryos that made bred animals, or patterns associated with embryos that did not produce pregnancies. I'll be honest, I don't know what these patterns are. Like I said, they're built off of millions of parameters, but the great news is they're highly predictive. And a question that we get asked a lot is, are your outcomes binary? Do you say that they are good or bad? And the answer is no. Typically, embryos aren't 100% good or 100% bad. There's definitely a variable category in there where they're alive, but they're not strong. And so how do we really give our users a good comprehensive evaluation of embryo's health? Well, like I said, a typical video is about 900 frames. So our computer will actually cast a prediction each frame. As you can see in this example, the computer might not predict the same prediction each frame. So this one for embryo 1, at first pass, it thought it was bred. Oh, frame 5, it didn't look so good, open, and then bred again. So just for this simplistic example, we can basically tally the predictions at the end of the video and express the outcomes as a percentage of total positive votes or total negative votes. So here we can get, that they are 80% likely to produce a pregnancy or 20% likely, rather than just a winner takes all prediction where it's either a good one or a bad one. And this is really important if we look at the landscape of our users. We work with many customers who work with very, very, very expensive genetics. Some of these embryos are worth thousands, if not tens of thousands, and sometimes even hundreds of thousands of dollars. They're probably not going to throw out an embryo that's worth $100,000. They still might want to know how likely it is to succeed, but even if it had only a 20% shot of making a pregnancy, it's probably worth it to them to at least try. On the flip side, there's companies making embryos very inexpensively, and sending them to Asian countries or African countries. These groups probably have more tolerance to discard embryos, so they'll probably only want to ship their clients customers that have 80% chance or better. So, presenting this data in a model like this puts the liability back on the user, and they can pair their own knowledge and know-how and use our score to manage their breeding strategy. And this is just the prediction to test our prediction accuracy. What we do is we see what the computer says. We do have to be binary when we're doing the testing stage. So we do that winner takes all and determine if the computer thinks it should produce a pregnancy or not. And then we compare it to the actual outcomes experienced after that embryo was transferred. And this is how we test our prediction accuracy. In this example, this shows that we were 83% accurate in guessing or predicting which embryos would make pregnancies. But in many of our studies, we've been up to 95% accurate in detecting which embryos are not going to make pregnancies. We're very thrilled with this statistic, and with more data, it should only improve from here. And this is just kind of a schematic of one of the studies that this data came from. I want to show you sort of a couple of examples, what's next. Thankfully, machine learning is very hot right now. So a lot of companies are spending billions of dollars to produce models and releasing them to companies at low cost. These are making it so much easier to access these models. And this is a demonstration of a new machine learning model released by Meta, Facebook's holding company. And if we look at this, you know, historically, segmentation was a very difficult and expensive process to create. And our machine learning programmer was able to use this tool and train it on embryos in less than an hour. Here is another example of a tool released by Meta. So right now, if we're getting 95% prediction accuracy, I can only imagine that the future in our ability to leverage other advancements in machine learning and artificial intelligence will only improve our ability to make quality, valid predictions in the future. So how can this be applied to the veterinarian? What we've seen so far is that, like I said in our early introductions, the average pregnancy rate is 50%. Without EmGenisys, practitioners say they have 100 available embryos. They would transfer all 100 into 100 recipients. With a typical 50% pregnancy rate, they're going to get 50 cows pregnant, 50 open. But if we used our technology to test these same 100 embryos, we would know that 20 were not viable. We would throw them out and stop wasting any more time and resources on them. Now they'll transfer just 80 embryos into 80 recipients. We'll get the same number of animals pregnant, but we can accomplish this with fewer costs and fewer animals. On the flip side, if they have embryos in frozen storage, say they still have 100 of recipients available, we can sub out embryos from frozen storage and actually use that to get more animals pregnant. So there's a lot of ways that this can be used, and they are advantageous to both the veterinarian and the cattle producer or animal breeder at the end of the day. Our goal is to use this to decrease the cost of ET and, if they choose to, reduce the size of the recipient herd. This really lowers the impact on the environment and on their bottom line. But we can also improve the producer's top line because they will increase overall net production. With a higher proportion of their herd, more of their animals will be making the money. If we're a dairy, animals have to get pregnant so they can lactate and make milk. We'll get these animals making milk sooner. And in both the beef and dairy situation, these embryos are typically higher value calves once they're born. Our company has tested well over 4,500 embryos at this point. And like I said, we've achieved about 85 to 95% prediction accuracy. We're fortunate to have users all over the world right now. And actually, I think we've worked with groups in every continent but Antarctica right now. So that's something that I will brag about and say very proudly. We have begun to collect data on 8 species, I believe. Cattle, sheep, goat, horses, pigs, bongo, kudu, Grevy's zebra, and humans. So actually 9. And we've even had offers to acquire our company. We talked about this in the beginning, but we've been very pursuant of getting quality IP and protecting our ideas. And so we're proud owners of 8 issued patents, with 7 others pending and 40 applications internationally. Exceptionally cool in our patent portfolio is we have protections on a claim of video analysis of embryos recorded at 2 frames per second or faster. In the patent world, broad is typically a good thing. So you can see the analysis is not explicitly defined, and a video at 2 frames per second is very slow. So essentially we have protection on any analysis that anyone could do on a video of an embryo, and also protection of these videos that are 10 minutes in duration or less, giving us the fastest real-time solution in the marketplace. And our goal is to create an investable and profitable company. But beyond that goal is we really do care about using technologies to promote human, animal, and environmental health and conserve the state of conservation. So all the species on this planet can not only survive, but thrive. We believe that by maximizing high quality genetics, we can really reduce disease, promote welfare, and create a sustainable and safe food supply for humans who are consuming our products. So we could not do this without our awesome team. So thank you so much, everyone, for being part of this. Andrew, this was awesome to get the opportunity to talk to you and your group. And with that, I have to say it because I always do. We are breeding something epic.
That is phenomenal. Early on, and just recently on one of these slides here, the word space appeared. Can you give us any hints of what does that mean? What's going on there?
I'd actually like to talk to you about that more in depth sometime, but the only thing that is conceived and given birth in space today is cockroaches, which why we would take cockroaches to space in a nice roach-free environment, that's beyond me.
So it was intentional is what you're saying.
Yes, I mean, groups want to know, can humans reproduce in space? Can mammals reproduce in space? It's never been done before. It will be impossible for us to colonize space and sustain populations if we can't reproduce. So something I believe in, or I would like to look at, is why hasn't a mammal successfully reproduced in space? You looked at my timeline. Hundreds of years ago, we could do IVF in rabbits, and here we are in 2023, and we can't get rabbits to breed in space. Why? So I don't believe that our technology can possess all the answers, but I do think that if we were to send mammalian embryos to a space-like environment, record videos of them, we can compare the real-time development of those space-produced embryos to the Earth-produced embryos and see what's different. We can then manipulate the space environment to be more Earth-like and see if we can get more Earth-like patterns in these embryos. So because we're the only group that can look at embryo development in real time, I think we can work on creating solutions that force embryos to be more Earth-like. So maybe one day it is possible for mammals, including humans, to reproduce in space. So that's a passion project of mine. I think it's fascinating. I think it'd be really cool to say that we're operating in that industry. So we're definitely looking for partners and collaborators who might want to go on that mission with us.
Yeah, you're right. We'll talk offline about this one.
But yeah, it's really cool. I think that there's a lot to be learned about early embryo development that can have a lot of application in a lot of different ways. And our goal is to bring these technologies to every industry possible so we can help as many people as possible.
Well, Kara, thank you. And you led me into my last question here is what can we do to help you? Both who is here, listening in live, but also the audiences that will view us later. What would you like to see us do for you?
Well, I mean, I think that since you work with so many startups, you know that our challenges are definitely getting people to know about us. I think we just described a ton of different applications, but we can't create solutions without partners to get us this data. We're also looking for customers. We have a technology that is delivering reliable results. And like Cameron said, we haven't launched our technology for anybody and everybody to use, but if a group approaches us, we can work with them to do the customizations necessary to launch that technology for their use. So any type of exposure, in my opinion, is beneficial to us. We're not experts in everything. If anybody has feedback, questions, concerns, advice, we are more than open to it. And, we're just really looking for people excited as we are to help bring ag innovation to the marketplace. So I know you have a lot of networks for that. You've already connected me to several of them, but, we want to hear as much feedback as possible and have as much exposure as possible.
Phenomenal. I really have nothing more to add. That was one of the best presentations I've seen. I assume you guys have practiced this one for hours prior to coming on this webinar. And if not, you mesh well together, that's for sure.
No, I'm lucky to have good backup. And no, I know that some of these slides were being modified this morning. So we work well under chaos.
Obviously. Does anyone have any questions for Kara or the team that's on the call right now? I know Cassie was having some connectivity issues. She had a few questions, but she's unfortunately unable to join us and hasn't texted me those questions yet. But we'll get to those. What else would folks like to add to this?
I appreciate the opportunity. Yeah, and please contact us, whether that's on social media, email, or through the contact form on our website. We'd be happy to address those questions or set up personal one-on-one Zoom calls if anyone's interested. But this was a great opportunity. Thanks for getting in the weeds with us with the data. And, we're proud of what we have, and we're happy to share what we know because we think that can just lead to opportunities.
Agreed. And thank you too. And as you know, I have your back covered. So thank you so much for coming on here and joining me for my webinar.
Appreciate it. Thank you so much.